So welcome everybody to our Immunology Pills mini webinar series with today's teaser presentation, Solutions to Enhance Your T-Cell Experiments. In the next 20 minutes, you will learn about Miltenyi Biotec's T-cell portfolio and how to best work with these important cells. For deeper and more extensive information, please visit our website and of course, contact your local sales organization. Please briefly acknowledge also the displayed disclaimer. Let's have a brief look at T cells and why they are so important. Well, T cells play as effectors of the immune system an integral role in the adaptive immunity. They can be subdivided broadly into several phenotypically and functionally distinct subsets, such as T helper cells, cytotoxic T killer cells, T regulatory cells, as well as the rather heterogenic group of unconventional T cells comprising, for example, natural killer T cells and gamma delta T cells. T cells are the only immune cells that are able to form a long-lasting immunological memory that helps to respond faster and more efficient to already experienced threats. Key surface markers are, of course, the lineage markers CD3 as well as CD4 and CD8, but also activation markers such as CD25, CD69, CD137, and CD154. T cells communicate and exert their functions mainly via cytokines such as interleukin-2, interleukin-4, TNF-alpha, and interferon gamma. T cells are thereby most relevant in both maintenance of health as well as fighting diseases. Last but not least, these properties make T cells ideal tools for immunotherapy, where, for example, T cells are genetically modified into CAR T cells to attack specifically cancer cells. Although our best-known products are indeed the microbeads and isolation kits, we are not just a cell separation company. We actually cover the whole experimental T-cell workflow from cell separation to cell analysis, including also sample preparation as well as cell culture. We provide thereby solutions for each step of your T-cell experiment. The best way to arrange maybe our quite large T-cell portfolio are distinct workflows, specifically designed for your experimental needs. Thereby, we cover as focus workflows, amongst many others, regulatory T cells, CAR T cell research, gamma delta T cells, tumor-infiltrating leukocytes, polarization of T cells, antigen and virus-specific T cells, and of course, activation and expansion of T cells. With these comprehensive workflows, you can easily choose the products relevant for your specific field of interest. Please note that our T-cell portfolio is growing by the day, and the respective workflows are all constantly evolving. So check regularly for updates. By the way, you might notice throughout the presentation these small yellow buttons paired with a little click hand symbol, just like the one on the lower right here. In the PDF version of this talk, you can access the respective product pages, marketing materials, videos, and protocols by simply clicking these buttons, as they are all hyperlinked. Well, getting your whole workflow from Miltenyi Biotec means ease of mind. Standardization, reproducibility, and organization of an experimental workflow become much more feasible. Miltenyi Biotec, as supplier for the whole T-cell workflow, means that all reagents fulfill the same high-quality criteria and the order process can be streamlined. Thereby, harmonizing the steps of an experiment as well as automation and upscaling are easily performed. Getting all products from Miltenyi guarantees smooth procedures and less errors, since, for example, our cell separation is tested for compatibility with our flow cytometry and our media for compatibility with our cytokines, and so forth. Moreover, having Miltenyi Biotec as full supplier of the whole workflow means also getting full support for every step of your experiment, thanks to our great scientific support team. Moreover, a hallmark feature of Miltenyi's workflow solutions is the seamless translation from research setting into a clinical setting and back, thanks to our MACSQuant portfolio, as well as cell manufacturing platforms such as the Clinimax Prodigy. So let's go to the main part of this presentation, our T-cell highlight products. Well, let's start with our journey through T cell workflow, taking the first step, sample preparation. Every successful experiment starts with a proper sample preparation. So start smart. Your sample needs to be prepared accordingly before starting your T cell experiment. This is especially true when working with human and mouse solid tissue. Keep in mind, the quality of sample preparation influences all downstream applications. For example, a homogeneous and viable single-cell suspension is the basis for successful and efficient cell separation. Miltenyi Biotec sample preparation solutions, namely the GentleMax tissue dissociator and the respective tissue dissociation kits, combine efficient automated mechanical tissue disruption with gentle and tissue-specific enzymatic digestion. With this, you obtain an optimal single-cell suspension with T cells of highest viability and preserved epitopes for the subsequent cell separation step. Streamlined sample preparation with the GentleMax ensures optimal reproducibility and downstream compatibility. The GentleMax hereby is programmable, has optional automated heating, and can process up to 8 samples in parallel, eliminating thereby user variability. Our sample preparation solutions are thereby rounded off with the tissue storage solution as well as the smart strainers and pre-separation filters to further optimize your workflow. Now, once your sample is properly prepared, it is time for the next step, the separation of your T cells. Of course, here our renowned MAX technology is the way to go. So select the best for all your cell separation needs. Cell separation poses actually many challenges to the researcher. High purity and recovery are of course important, but also reproducibility, cell viability, and compatibility to downstream applications have to be ensured. Furthermore, there is a variety of different T cell subtypes that might be of interest and have to be separated individually. Some of them display very low frequencies in the variety of different starting materials and are hard to obtain. And of course, cell separation should be fast and easy to use. Consequently, there are many cell separation challenges to face, and gladly Miltenyi Biotec offers the right solutions for all of them. Generally, MAX cell separation technology offers two different separation strategies. First, positive selection with our microbeads and microbead kits. Target cells are hereby labeled and directly selected via their specific and defining marker. As target cells are still magnetically labeled afterwards, they are usually not suited for an additional round of separation, except when you use release microbeads, where the label can be removed via an additional step. The positive selection approach with microbeads is very specific and therefore ideally suited for rare cells or undefined single-cell suspensions, such as tumors and other tissue, as well as difficult or compromised samples, for example from sick donors. Also, isolation directly from whole blood and blood products is very well achieved with MAX positive selection. Second, we have the untouched isolation with our isolation kits, working via the depletion of non-target cells. This is necessary when the target cell fraction is not defined by a certain marker, but rather by the lack thereof. Hereby, all cells expressing defined exclusion markers are depleted and the target cells remain unlabeled and thus are usable for further rounds of sub-separation. However, in order to work efficiently, the single-cell suspension has to be rather well defined, as it is the case with, for example, human PBMCs or mouse spleen single-cell suspensions. Taken together, different isolation strategies allow to get the most out of your sample, with Miltenyi being an expert in all cell separation strategies, playing with and combining the different approaches offers a great flexibility according to your needs and is the secret to success. But why use Miltenyi Biotec for cell enrichment and what does make MAX cell separation technology so special? Well, in short, it is our select the best approach. Minimal labeling with no effects on cell biology for full downstream compatibility. In more detail, our MAX columns are a technological advantage. The column amplifies the magnetic field by 10,000-fold, and this allows for minimal labeling and smallest bead size of only around 50 nanometers. In consequence, less epitopes are covered on the cell surface, and less stress is applied on the cells. So, as thousands of publications prove, the isolated T cells are fully suited for any downstream application, including flow analysis. Thereby, MAX microbeads were even approved by the FDA to be used in specific clinical settings. It is important to note that the column allows for the most gentle sorting, since there is a large space between the steel spheres of the column matrix with about 20 times the size of a lymphocyte. The T cells will pass through smoothly without any negative effects. Have a look at the data on the right side. We see these relabeled T cells. Column-free technology results in bead aggregates and massive overlabeling on the cell surface. With MAX column technology, you see no bead aggregates and minimal labeling for fully functional cells. Consequently, MAX cell separation technologies comes with 4 different approaches to perfectly reflect the different starting materials. First, we have microbeads and microbead kits that are ideal for positive T cell separation from PBMCs and various tissue single cell suspensions. They also include the new REALise technology for label-free positive cell separation. Separation of basic T cells and almost any T cell subtype from different species like human, mouse, rat, and even non-human primate make microbeads and microbead kits a very versatile cell separation approach. Second, we have isolation kits that are ideal for all untouched separation needs. The used starting materials are PBMCs or defined single-cell suspensions. Also, the isolation kits cover basic T cells as well as almost any T cell subtype from different species like human, mouse, rat, and again non-human primate. However, if you do not want to go over density gradient to get PBMCs, for example, as starting material, you can also isolate T cells fast and directly from whole blood and blood products. So as a third option, we have our straight from microbead kits for the isolation of human CD3, CD4, and CD8 T cells directly from whole blood and blood products without the need for density gradient. It is a reliable way to get your T cells fast and easy and is of course fully automatable. As fourth separation variant, we have offer our MACSQuant isolation kits. Also here, human Tregs, PAN T cells, as well as CD4 and CD8 subsets and even CD4 memory T cells can be isolated directly from whole blood and blood products without the need for density gradient. This time, however, it is an untouched cell isolation approach. MACSQuant is thereby the fastest and most uncomplicated manual cell separation approach. One of the many things that sets Miltenyi's T-cell portfolio apart is its broadness and diversity. We cover more than just the basic human and mouse T-cell subtypes. Indeed, over 90 T-cell separation products cover virtually every T-cell subset, addressing T-cell researchers' needs worldwide. For example, different naive and memory T-cell subsets, as well as T helper cell subsets, are covered. Furthermore, different T regulatory cells, gamma delta T cells, and NK T cells separation kits are available, as well as complete workflows for antigen-specific T cells, cytokine secreting T cells, and CAR T cell research, and of course, many, many more. Apart from human and mouse samples, we also offer T cell separation kits for rat and non-human primate samples. As scientists, we understand how important it is to address the full complexity of T cell subtypes, even for the rare ones, and to offer according solutions. MAX technology offers, depending on the researcher's need, different manual and automated cell separation options for upscaling. Manual separation with the Quadro or OctoMax for up to 4 or 8 samples in parallel, respectively. Automated options include the AutoMax Pro for full walkaway convenience for up to 5 samples. The Multimax Cell 24 Separator Plus for the processing of up to 24 samples in parallel. And finally, the Multimax X for flexible, fully automated, high-throughput industrial applications with any source material. Now, every researcher can reliably automate and parallelize cell separation for more efficient workflows and higher consistency. Of course, the automation options are applicable for most of our T-cell separation solutions. Well, now for the next step. We successfully isolated T cells. It is time for the culture step of your experiment. So cultivate your vision. When culturing T cells, apart from activation and expansion, also differentiation into different T cell subtypes is an important topic. The latter is performed, for example, with our MAX cytokines and functional grade antibodies. Activation is performed with either polyclonal stimulation reagents that, for example, engage CD3 and CD28, like our innovative T-cell transect nanometrics, or antigen specifically, using, for example, peptivator peptide pools. Expansion and culturing is, of course, best performed in special T-cell media, such as our serum-free Tex-Mex. As you can see, Miltenyi Biotec hereby covers the full range of T-cell culture from media to cytokines and from activation and expansion to polarization. Our next part then covers T-cell analysis. The method of choice is of course flow cytometry for assessing T-cell surface markers, secreted cytokines and transcription factors. Miltenyi's extensive flow analysis portfolio does not only include antibodies, but also flow kits, cytometers, flow sorter, and even imaging systems. Thereby, every researcher's need is addressed. So join the flow revolution. Maybe the highlight product in the flow chapter are the REAfinity Recombinant Antibodies. Being recombinantly engineered, we have full control over their genetic sequence and can express the REAfinity antibodies in mammalian non-hybridoma cell systems. This means that REAfinity antibodies exhibit highest purity and lot-to-lot consistency for greater reproducibility, that they can undergo the most stringent in-house validation, and there is no need for tedious and costly Fc receptor blocking. And they require only one universal isotype control for maximum convenience and again cost saving. REAfinity antibodies are hereby readily available in hundreds of specificity and are best combined with our super bright proprietary bio-dye fluorescent antibody conjugates. Most recently, even releasable versions of for label-free cell sorting were added to our REAfinity portfolio. Additionally, REAfinity antibodies also are parts of our unique Miltenyi Biotec tested panels, various detection kits, and pre-mixed cocktails, including the innovative stain-expressed dry antibody cocktails. In this context, especially our ready-to-use, standardized, and convenient phenotyping and cytokine-secreting cell analysis kits will make your life so much easier. More info on that can be found by following the various blue links on this slide. Our flow portfolio is rounded off with our flow analysis and sorting instruments of the MACSQuant series. First, we have the MACSQuant Analyzer, powerful benchtop flow cytometers with up to 16 optical parameters. The clue here is that they allow for full automated acquisition using a robotic sample collector, and with the express mode software modules, even data analysis is automated and free of human error. The small benchtop cytometers fit easily under the flow hood, adding further to operator safety, for example when working with contaminated samples. Second, we have the MACSQuant Taito cell sorter. Opposed to classical droplet sorters, the MACSQuant Taito allows for the most gentle sorting and highest viability even for rare and fragile cells, thanks to its unique microchip sorting technology. Furthermore, the closed and sterile cartridge system makes the Taito the ideal machine for safe GMP environment. Moreover, a really fantastic novelty in our portfolio are our new imaging systems, MACSIMA and the UltraMicroscope Light Sheet Microscope. These systems allow the visualization of T cells and others within a tissue context. Thereby, new CAR T targets may be identified, the distribution dynamics of TILs in tumor tissue assessed, and a deep phenotypic analysis of donor samples and the tumor microenvironment is possible. For once, the MAC-Sima Imaging platform for ultra-high content imaging thereby applies the renowned MIX technology. Spatial immune cell distribution, including their phenotypes, is assessed within tissue and cell conglomerates by the sequential stainings of hundreds of markers. In contrast, traditional staining techniques allow maybe the staining of 3 markers, giving only an incomplete picture. This analysis of hundreds of markers within a single sample includes, of course, automated sample processing and imaging of all types of fixed samples, from tissue to single cells. Have a look at the product video by clicking the respective links on the left. On the other hand, the ultramicroscopes of the UltraMicroscope family enable you to visualize whole biological systems in 3D. Moving the sample through the light sheet allows the generation of sharp and bright 3D images of, for example, the tumor microenvironment, thereby giving you full insight into the tumor immune cell interactions. Please have a look at this impressive video here that was taken with the UltraMicroscope Blaze that shows a mouse xenograft of a human pancreatic carcinoma cell line with infiltrating CAR T cells in 3D. Although our T-cell portfolio is quite extensive, as you can see here in this full T-cell workflow overview, you have to remember just 3 easy things. First, we offer more than just T-cell separation. Second, we cover thereby your whole T-cell workflow. And third, we have a solution for every pain point. So have all the success with our innovative and complete T-cell portfolio, as displayed here in the full-featured overview. Our T-cell portfolio is growing by the day, and the respective workflow overviews are all constantly evolving. So please check for regular updates. Please follow also the links on the lower right to access the respective T-cell product pages, as well as to download our T-cell brochures. Talking about more information, of course, you can find a variety of helpful marketing material downloadable from your webpage, www.miltenyibiotec.com. For T-cell specific applications, just click on the button on the lower right side. There you will find flyers and brochures, webinars and videos, protocols and datasheets, customer reports, application notes, scientific posters, and datasets. And of course, you can always directly reach out to your local sales organization for deeper and more extensive information. Hereby I close with this nice picture of our company campus in Bergisch Gladbach at night. Please don't forget to visit our website and to contact your local sales organization. Thank you very much for your attention.